Hey everyone, imagine taking a cell and placing it into pure water. Water would begin moving into the cell through osmosis. As pressure inside the cell increased, the membrane alone might not be strong enough to resist that force. For many cells, the result would be swelling, deformation, or even rupture. So, how do microbes survive in environments where water and solute concentrations are constantly changing? One major answer is the cell wall. The cell wall provides structural support and protection outside the plasma membrane. It helps cells resist osmotic pressure, maintain shape, and survive environmental stress. And although many organisms have cell walls, not all cell walls are built the same way. In this lecture, we will explore the structure and function of microbial cell walls and why they are so important in microbiology. By the end of this lecture, you should be able to explain how cell wall structure helps microorganisms maintain shape and survive osmotic stress, and compare major microbial cell wall types, including gram-positive, gram-negative, acid-fast, and archaeal cell walls. Pardon me. The cell wall is a rigid structure located outside the plasma membrane. One of its most important functions is maintaining cell shape. Without the support of the cell wall, many bacteria would not be able to maintain their characteristic morphology. The cell wall also protects against osmotic rupture. Because cells often contain higher concentrations of solutes than their surrounding environment, water frequently moves into cells through osmosis. This creates internal pressure, sometimes called turgor pressure. The rigid cell wall helps resist that pressure and prevents the cell from bursting. Cell walls are extremely common in prokaryotes, including most bacteria and many archaea, although their exact structure can vary between groups. So, while the plasma membrane forms the selective living boundary of the cell wall provides structural support and protection. The main structural component of most bacterial cell walls is peptidoglycan. Peptidoglycan is a strong mesh like polymer that surrounds the outside of the cell membrane. What makes peptidoglycan especially important is that it is unique to bacteria. Neither archaea nor eukaryotic cells contain true peptidoglycan. The structure is built from repeating sugar molecules called NAG and NAM that are linked together into long chains. These chains are then connected by short peptides, creating a strong network surrounding the cell. This mesh like structure provides strength and rigidity to the bacterial cell wall. It helps maintain cell shape and protects the cell against osmotic pressure that could otherwise cause rupture. Because peptidoglycan is unique to bacteria and essential for survival, it is also an important target for many antibiotics. Now, gram-positive bacteria have a relatively simple but very thick cell wall structure. Their most noticeable feature is a thick layer of peptidoglycan surrounding the plasma membrane. This thick peptidoglycan layer provides structural support and helps protect the cell from osmotic stress. Unlike gram-negative bacteria, gram-positive do not have an outer membrane. The peptidoglycan layer is the outermost major structure surrounding the cell. Gram-positive bacteria also commonly contain molecules called teichoic acids embedded within the cell wall. These molecules contribute to the cell wall stability and may play roles in cell interactions and ion balance. The thick peptidoglycan layer is also important during gram staining. When cells are stained with crystal violet and then treated during the staining process, gram-positive cells retain the crystal violet dye and appear purple under the microscope. This staining difference is one of the most important ways microbiologists classify bacteria. Gram-negative bacteria have a more complex cell envelope structure than gram-positive bacteria. Instead of a thick peptidoglycan layer, gram-negative cells have a relatively thin layer of peptidoglycan located between two membranes. Outside the plasma membrane is an additional outer membrane that surrounds the cell. This outer membrane contains lipopolysaccharide, also known as LPS. It's an important molecule found only in gram-negative bacteria. LPS can help protect the cell and also plays important roles in interactions with hosts and our immune system. Because gram-negative bacteria possess both an outer membrane and a thin peptidoglycan layer, their cell envelope is structurally more complex than that of gram-positive bacteria. The outer membrane can also act as an additional barrier, making some gram-negative bacteria more resistant to certain antibiotics and environmental stresses. Some bacteria have cell walls that are even more unusual. Acid-fast bacteria contain large amounts of a waxy molecule called mycolic acid in their cell walls. These lipids create a thick hydrophobic outer layer that makes the cell wall highly resistant to drying, chemicals, and many environmental stresses. Because of this waxy structure, acid-fast bacteria do not stain well using the traditional gram stain. Instead, they require a special acid-fast staining procedure that allows the cells to retain certain dyes even after treatment with acidic solutions. One of the most important groups of acid-fast bacteria is the genus Mycobacterium, which includes species that cause diseases such as tuberculosis and leprosy. So, although acid-fast bacteria are structurally related to gram-positive bacteria, their unusual waxy cell walls give them very different staining properties and environmental resistance. Although cell walls are extremely important for many microbes, some bacteria can survive without them under certain conditions. These wall-deficient cells are called L-forms. L-forms may arise when bacteria lose their cell walls because of mutations, environmental stress, or exposure to antibiotics that target the peptidoglycan synthesis. Without a rigid cell wall, these cells become much more sensitive to osmotic stress because they no longer have strong structural protection against internal pressure. As a result, L forms often display irregular or variable shapes instead of the more defined morphology seen in typical bacteria. Some microorganisms also naturally lack cell walls entirely. For example, members of the genus Mycoplasma do not produce a typical bacterial cell wall at all. These organisms rely on other structural adaptations to survive and maintain membrane stability. So while cell walls are common and extremely important in microbiology, they are not universal. Bacteria are often divided into two major groups based on differences in cell wall structure: gram-positive and gram-negative cells. Gram-positive bacteria have a thick layer of peptidoglycans surrounding the plasma membrane. They do not possess an outer membrane. Gram-negative bacteria are different. They have a much thinner peptidoglycan layer located between the plasma membrane and an additional outer membrane. These structural differences are important because they influence how cells interact with their environment. They also affect staining behavior during the gram stain procedure. Gram-positive cells retain crystal violet stain and appear purple, while gram-negative cells do not retain the stain and instead appear pink after counterstaining. The structure of the cell envelope can also influence resistance to environmental stress and antibiotics. For example, the outer membrane of gram-negative bacteria can act as an additional protective barrier. So although both groups are bacteria, their cell envelope structures are fundamentally different. Archaea also possess cell walls, but their walls are fundamentally different from those of bacteria. One major difference is that archaea lack true peptidoglycan. Instead, archaeal cell walls can have a wide variety of compositions depending on the species. Some archaea contain a material called pseudopeptidoglycan, which is similar in function to bacterial peptidoglycan, but chemically distinct. Others may use proteins or different polysaccharides to build their cell walls. These alternative wall structures help archaea survive in a wide range of environments, including some of the most extreme conditions on Earth. Many archaea live in environments with very high temperatures, extreme salinity, or high acidity. Their unique cell wall chemistry contributes to the stability and protection needed to survive under these harsh conditions. Cell walls are much more than simple outer coverings. They help cells maintain their morphology and resist osmotic pressure that could otherwise damage or rupture the cell. Cell walls also play major roles in protection and environmental survival. In microbiology and medicine, cell walls are especially important because they are common targets for antibiotics. Many antibiotics work by disrupting peptidoglycan synthesis, weakening the wall and causing bacterial cells to fail under osmotic stress. Cell wall structure can also influence ecology and pathogenicity. Differences in cell envelopes affect how microbes interact with hosts, survive environmental stress, and attach to surfaces, and respond to antimicrobial compounds. So far, we've mainly focused on structures that surround the cell, the membranes, and the cell wall, but cells also contain many important internal structures that carry out metabolism, store information, and support survival. In the next lecture, we'll move inside the cell and explore the internal structures of prokaryotic cells. With that, thanks for listening to this lecture. See you next time.